I want you to try something for me, just for a second. Close your eyes and just imagine your heart beating. Just one beat, thump, thump. It feels simple, doesn't it?
It does.
It feels like one cohesive, singular event. A muscle squeezes, blood moves, and that's the end of the story.
That's certainly how it feels to the user. I mean, it's the clean user interface version of our bodies.
The user interface, I like that.
But as we all know, the UI is really just there to hide the insane complexity of the code that's running underneath.
Exactly, because if we zoom in, and I don't just mean to the tissue level, I mean if we really, really strip this down to the microscopic reality, that single thump isn't a singular event at all.
Not even close.
It is a frantic, high-stakes negotiation. It's billions of tiny walled city-states all agreeing to open their gates at the exact same millisecond, flooding their streets with ions, burning fuel, and ratcheting these microscopic gears.
And they're doing all of this while being under constant siege. We tend to think of homeostasis, that perfect balance our body keeps, as a peaceful state.
Right, like it's just sitting there calm.
But it's not peace. It's a dynamic, moment-to-moment standoff. It's an active process.
Yeah, we tend to walk around thinking of ourselves as these solid, singular beings. I am a person. But biologically, we're really just a collection. We're a galaxy of trillions of cells. And today, we're going to look at just one of them.
We are officially stepping away from the bag of jello model.
Oh, the high school biology circle with the dot in the middle.
Gone. We're throwing it out.
Oh.
That model did us a huge disservice because it taught us the names of the parts, but it never taught us the logic of the system.
And that's what we're here for.
Today, we are treating the cell as what it actually is. A bustling, dangerous, highly regulated, microscopic metropolis. This is our deep dive into cellular physiology.
And to guide us through this city, we aren't just skimming the surface. We have pulled two of the absolute heavyweights of medical literature off the shelf. We're going to be synthesizing Ganong's review of medical physiology and the legendary Guyton and Hall textbook of medical physiology.
The Bibles, as they're known in pretty much every med school. And honestly, you can't really get a deep understanding of physiology without grappling with both of these texts.
It's fascinating because as we were prepping for this, we realized that these two authors, these two traditions, they look at the exact same microscopic world, but they see two very different things.
They really do.
How would you characterize that difference for everyone listening?
Okay, so think of Ganong as your master mechanic, or maybe a chemical engineer. Ganong is obsessed with the nuts and bolts.
Okay.
When you read Ganong, you aren't just learning that a cell signals, you're learning the precise molecular geometry of the receptor, the interfaces, the chemical specificity. It's the how at the most granular protein-to-protein level.
So Ganong is the guy with the magnifying glass, and he's looking at the individual weld joints on the steel beams.
Precisely. Now, Guyton. Guyton is the architect.
Oh,
Or maybe the city planner. Guyton acknowledges the bolts, of course, but he is obsessed with the flow. He wants to know about traffic patterns, water pressure, the energy grids. He focuses on systems and this concept he calls quantitative intuition. He really wants you to feel how the cell fits into the whole human.
The mechanic and the architect. And our mission today is to try and mash these two perspectives together. We want to build a conceptual map of the cell that has the mechanical precision of Ganong, but also the systemic beauty of Guyton.
Right, and I should probably add a quick disclaimer here, though. We are synthesizing thousands of pages of dense text into, an hour or so?
Something like that.
This is a framework. Think of what we're doing today as building the skeleton. You still have to go read the texts yourself to put the meat on the bones. Reading is absolutely non-negotiable if you want mastery.
That is a fair warning. Now, before we open the gates to this city, there is a theme that just kept hitting me in the face while I was reading these chapters. A central tension.
Regulation versus capacity.
It's everywhere, in every single part of the cell.
It feels like the fundamental struggle of being alive, doesn't it?
Let's unpack that a little bit. What do we actually mean by regulation versus capacity?
Okay, think about it like this. To survive, a cell needs capacity. It needs to be a powerhouse. It needs to burn energy, move massive amounts of material, replicate its DNA, react instantly.
That is throughput.
Exactly. It needs to be able to do things on a massive scale.
But if you just have unchecked capacity...
You have chaos. You have a nuclear meltdown. In biological terms, you have cancer or you have autolysis, which is self-digestion. So you need regulation. You need these incredibly tight constraints to maintain homeostasis. You have to hold back all that power.
So it's like trying to drive a Formula One race car at 200 miles an hour. That's the capacity. While trying to balance a full cup of hot coffee on the dashboard. That's the regulation.
And the absolute miracle of biology is that most of the time, the coffee doesn't spill. That tension is what we are going to explore in every single organelle we discuss today.
I love that. So let's start at the edge, the border control. We are entering section 1, the barrier and the gatekeepers, membrane physiology.
The city walls.
And again, I have to blame my 10th grade biology class here. I always pictured the cell membrane as basically a Ziploc bag.
Yeah.
You know, a piece of plastic that just keeps the inside stuff in and the outside stuff out.
It's a very static view, and it's one Guyton just destroys pretty much right away. He starts with the most basic element, water.
Right, because we are mostly water.
We are. The cell is 70 to 85% water. The stuff inside, the protoplasm, if you want to use the old school term, it's basically just water with salts, proteins, and electrolytes dissolved in it.
And the outside of the cell, the extracellular fluid, is also water.
Also water.
So if I have water on the inside and water on the outside, why don't they just mix? I mean, why doesn't the cell just dissolve away like a sugar cube?
Because of the fat sandwich.
The lipid bilayer.
Yeah, exactly. This is the magic of chemistry. You have these molecules called phospholipids. They're amphipathic, which is just a fancy way of saying they have a split personality.
Okay.
One end, the phosphate head, loves water. It's hydrophilic. The other end, the two fatty acid tails, is terrified of water. It's hydrophobic.
So when you throw a billion of them into a watery environment, they basically panic.
They self-organize. It's just thermodynamics at work. The tails all hide from the water by pointing inward at each other, and the heads bravely face the water on both the outside and the inside.
So they create a double layer.
A sandwich, where the bread is the water-loving heads, and the filling is pure water-hating fat.
And since water-soluble things like sugar or ions or urea can't pass through a layer of fat, you've suddenly created a perfect seal.
It separates the inside fluids from the outside fluids. But here's where that regulation versus capacity tension kicks in immediately.
Right, because if it were just a seal, nothing could get in. The cell would starve.
It would be a tomb. So this wall needs to be dynamic. This is the famous fluid mosaic model.
Fluid being the key word there. The wall isn't solid concrete. It's more like a sea of oil.
Yes. The individual lipids are constantly moving, they're shifting, they're dancing around each other. And this is where we meet a molecule that, often gets a bad rap. Cholesterol.
Yeah, we usually hear cholesterol and we immediately think heart disease, clogged arteries.
We do. But at the cellular level, cholesterol is an absolute hero. It's the regulator of that fluidity. Think of cholesterol molecules as these stiff little stakes that are driven down into that oily membrane.
Okay, so what's their job? What are they doing in there?
Temperature control. They're thermal buffers. Imagine if your body temperature goes up, say you have a fever or you're exercising hard. The lipid membrane naturally wants to melt. It wants to get too runny, like butter on a hot skillet.
Which would mean the cell just falls apart and leaks everywhere.
Right. So cholesterol grabs onto those neighboring lipids and says, whoa, hold on, stay together. It limits their movement so the wall doesn't collapse.
But what about the opposite? What if it gets cold?
If it gets cold, the lipids want to pack together and essentially freeze into a solid chunk of fat. Cholesterol gets in between them, physically preventing that tight packing. It says, no, keep moving, stay fluid.
So it prevents the wall from becoming brittle.
It's the Goldilocks molecule. Not too hot, not too cold. It keeps things just right.
That's incredible.
Without cholesterol, your cells would either rupture from heat or freeze solid. It is the stabilizer that allows the membrane to function in a constantly changing environment.
Now, Gannon points out that this sea of lipids is really just the background. A wall is pretty useless if you don't have doors and windows. We need to talk about the proteins.
The machinery, yeah. If the lipids are the drywall, the proteins are the doors, the windows, the security cameras, and the border patrol agents. In fact, by mass, the membrane is about 55% protein.
Whoa, that's way higher than I would have expected. More than half the wall is actually machinery.
It's a very busy place. And we generally divide them into two main camps. You have your integral protein and your peripheral proteins.
Okay, so integral must mean they go all the way through the membrane.
Correct. They span the entire thickness of the membrane. These are your channels, your pumps, your pores. They are the tunnels that allow specific things to bypass that fat barrier.
So this is how something like glucose gets in.
Exactly. Glucose is water-soluble. It can't just walk through the fat wall. It needs a VIP entrance. An integral protein forms a channel specifically shaped for glucose and ushers it through. That's capacity getting the fuel in.
But the regulation part is that the door only opens when, say, insulin tells it to.
Precisely. Now, what about the peripheral proteins?
They don't go all the way through, I'm guessing.
Right, they're usually attached to one side, very often on the inside surface.
Yeah.
They function more like controllers. Imagine a security guard who's sitting inside the lobby, and he's holding the remote control for the automatic door.
So that's a peripheral protein regulating an integral protein.
You got it. They're often enzymes or controllers for the pores and channels.
Guénon gets really specific here with something he calls a GPI anchor.
The glycosylphosphatidylineacetyl anchor.
Yeah, that. Let's just stick with GPI. Why does Guénon make a point to highlight this specific thing?
It's all about specificity and location. Sometimes the cell needs to station a protein on the outside surface, like a sensor or an enzyme that needs to work on the exterior. But proteins don't float around in lipids very well. They might just drift away.
So the GPI anchor is like a molecular tether.
That's a perfect way to put it. It roots the protein into the lipid layer so it stays exactly where it's needed.
It's like bolting a satellite dish to the roof of a building so it doesn't blow off in the wind.
Exactly. It just shows the incredible level of architectural planning that's involved here.
And this leads us to the final layer of the wall, kind of the paint job, if you will, the glycocolex.
Which sounds like a villain from a sci-fi movie, but it's actually just a sugar coat.
A sugar coat on the outside of the cell.
Yes. Most of the proteins and even some of the lipids on the outer surface have these little chains of carbohydrate sugars dangling off them. It creates this loose, fuzzy coat around the entire cell.
And this isn't just decoration, right? This is the cell's ID badge.
It's the ID badge, the force field, and the passport all in one. First, it has a net negative electrical charge.
Okay, why is that important?
Well, that charge repels other negatively charged objects. It's one of the reasons your red blood cells don't just clump together in your bloodstream. It enforces a kind of social distancing.
Social distancing for cells, I like that.
But more importantly, it's the passport. This is how your immune system tells self from foreign. Your white blood cells are constantly bumping into that sugar coat. If the pattern is correct, they say, okay, you're a civilian, you belong here, carry on.
But if the pattern is wrong...
If the pattern is wrong, like on a bacteria or a virus or mismatched blood transfusion, they attack.
So the membrane isn't just a bag. It's a dynamic fluid, temperature-regulated, heavily guarded border with a very strict ID policy.
It controls the who, the what, and the when of everything that enters the city. And that control is the absolute first layer of regulation in our theme today.
Okay, we've made it past security. We are now inside the city, which means we're moving to Section 2. The structural framework. The cytoskeleton.
Welcome to the cytoplasm.
Now, I have to correct another misconception I had here. I always thought the inside of a cell was just goo, protoplasm. Like if you could poke it, it would just squish.
The soup theory. Yeah, it's completely wrong. If you could shrink down and stand inside a cell, you wouldn't be floating in soup. You would be standing in a dense, almost terrifyingly complex construction zone.
So I'd be looking at a scaffold.
A massive 3D lattice of beams, ropes, and highways crisscrossing in every direction.
And Guénon makes a point that I absolutely loved about this.
Yeah.
This scaffold is not static. It's not like the steel beams of a skyscraper that get bolted in and then just stay there for 50 years.
No, it's incredibly dynamic. It is constantly assembling and disassembling. Imagine a construction crew that builds a bridge just for one specific truck to cross. And as soon as that truck is across, they rip the bridge apart behind it and run to the front to build the next section of the bridge.
That sounds like wild efficiency or inefficiency, I can't decide.
It's necessary. If the skeleton were rigid, the cell couldn't move. It couldn't divide. It couldn't adapt to its environment.
Okay, so let's break down the 3 pillars of this support system. First up, microtubules.
The highways. These are long, hollow tubes made of a protein called tubulin, specifically alpha and beta tubulin subunits.
And these are the ones that have that dynamic instability we were just talking about.
Yes. They are polar. They have what's called a plus end and a minus end. They can grow rapidly at the plus end and then shrink catastrophically at the minus end. This allows the cell to change its shape or redirect traffic almost instantly.
And what do they actually do besides just growing and shrinking?
They are the main tracks for intracellular transport, which we'll get to in a minute, but they also form the mitotic spindle. When a cell needs to divide, it builds this massive, intricate machine out of microtubules to physically pull the chromosomes apart into the two new daughter cells.
And this is where we get a really cool clinical link. The chemo drug, Taxol. How does that work?
Taxol or paclitaxel is fascinating. We usually think of killing a cancer cell by, blowing it up or poisoning it somehow. But Taxol kills cancer cells by making them too stable.
Wait, making them more stable kills them. How does that work?
It freezes the microtubules, it binds them, and prevents them from disassembling. Now remember, to divide, to perform mitosis, a cell has to completely tear down its existing skeleton and build that mitotic spindle from scratch.
So if the cancer cell is full of Taxol,
It can't tear down the old skeleton. The parts are glued together. It tries to divide. It hits this concrete wall of frozen microtubules, and panic ensues. The cell realizes it's stuck, that something is catastrophically wrong, and it triggers apoptosis. It commits suicide.
we literally gridlock the city to death.
That is exactly it. We use the cell's absolute need for dynamic regulation against it.
That's incredible. Okay, pillar number 2, microfilaments.
The muscles of the cell. These are made of a protein called actin. And actin is everywhere. I mean, in a typical mammalian cell, up to 15% of the total protein mass is just actin.
15%. That's a huge amount of resources dedicated to just one protein. What's it all doing?
Well, it provides the structural stiffness right under the cell membrane, what's sometimes called the ectoplasm. But its main job is movement. When a cell needs to crawl, it's using actin. When a muscle contracts, it's actin interacting with myosin.
We'll talk about the crawling in a bit because it is, frankly, nightmare fuel. But first, the third pillar, intermediate filaments.
The ropes. If microtubules are for resisting compression like pillars and actin is for generating tension like cables, then intermediate filaments are for providing shear strength. They keep the cell from tearing apart when it's stretched.
And these are different depending on where you look in the body.
Yes, and this is that specificity again. In your skin cells, they are made of keratin. That's why your skin is so tough and resilient. In your nerve cells, they are called neurofilaments. They are the reason you can stretch your arm and your long, skinny nerve axons don't just snap in half.
So we have highways, muscles, and ropes. But a highway is totally useless without vehicles on it. We need to talk about the molecular motors.
The trucks.
The trucks.
This is, hands down, my favorite part of cellular biology. If you ever look at an animation of this process, you will swear that it's fake. These are proteins that physically walk along the microtubules.
They look like little stick figures with big feet, and they're carrying these massive balloons over their heads.
They do, and they take literal steps. One protein foot, then the other. And they burn one molecule of ATP for every single step they take. It's incredible.
And we have two main trucking companies in here, kinesin and dynein.
That's right. And they have different routes, generally speaking. The cell has a center, usually near the nucleus where the microtubules originate, and the suburbs, which is the periphery near the membrane.
So,
The microtubule is laid out with its plus end pointing out towards the suburbs. Kinesin is a plus end directed motor, so it walks toward the plus end. It carries cargo out to the suburbs.
and dynein, I assume, goes the other way.
Dynein moves toward the minus end. It brings cargo in from the suburbs toward the city center.
Okay, so let's say a virus manages to break into the cell. It lands in the suburbs, near the membrane. But it needs to get to the nucleus to hijack the command center.
It looks for a dynein truck. It literally hijacks the cell's own transport system. Some viruses have evolved to have receptors on their surface that mimic the cell's own cargo, so they can hitch an express ride straight to the nucleus.
That is terrifyingly smart. It's a constant arms race.
The cell builds a beautiful, efficient highway for its own logistics. The enemy immediately figures out how to use it for an invasion.
Okay, let's follow those trucks. Where are they going with all this cargo? We are now entering Section 3, the manufacturing and logistics districts. Organelles,
And we have to start at headquarters, the nucleus,
the library.
the library, and the command center. This is where the master blueprints are stored, the DNA. And Guyton emphasizes that the nucleus is the control center, not because it does the work itself, but because it determines which work gets done and when.
It issues the work orders.
Exactly. And the security here is even tighter than at the outer membrane. The nuclear pores are incredibly complex gates. They will allow RNA, the temporary copies of the orders, to leave, and they'll allow specific proteins, the tools and machinery, to enter, but they will never, ever let the original DNA leave.
The master blueprints must stay in the vault at all times.
Always. Now, inside the nucleus, there's usually a dense, dark spot that's called the nucleolus.
The factory that builds the other factories.
That's the best way to describe it. Before you can build a protein, you need a machine to actually build it. That machine is called the ribosome. The nucleolus is the site of ribosome synthesis.
So the ribosomes get built in the HQ, they get shipped out through the nuclear pores, and they land in the assembly line, the endoplasmic reticulum, or the ER.
The ER is a massive organelle. It's this huge network of interconnected tubules and sacs with a surface area that can be 30 to 40 times larger than the cell membrane itself. It's all folded up and packed in there very tightly.
And we have two different flavors of it, rough and smooth.
Right. The rough ER is rough because it is studded with all those ribosomes we just made.
It looks bumpy under a microscope.
Exactly. This is where protein synthesis happens. The ribosome grabs an RNA work order, it reads the code, and it extrudes a brand new protein directly into the ER matrix. It's like a 3D printer injecting plastic into a pre-made mold for folding.
And the smooth ER. No bumps.
Yeah, no ribosomes. So it's not making proteins, it's making lipids, fats, and steroids. And crucially, it is the primary site of detoxification in the cell.
And this is a really big deal in the liver, right?
Huge. If you go out for a night of drinking or you take a medication like Tylenol, the smooth ER in your liver cells goes into overdrive. It contains a whole host of enzymes that modify these toxins to make them water-soluble so your kidneys can filter them out of your blood.
And there's a capacity adaptation here, isn't there? A change in the city layout based on demand.
Absolutely. If you are a chronic drinker, for instance, your liver cells physically expand their smooth ER. They build more factory floor to handle the increased toxic load.
So capacity increases to meet the demand.
It does, but it comes at a cost, of course. It takes up space and a lot of energy to maintain all that extra machinery.
Okay, so the ER makes the protein, but it's raw, it's unfinished, it needs to be polished, painted, and most importantly, addressed for shipping. That's the job of the Golgi apparatus.
The post office of the cell. The Golgi is a stack of flattened sacs, kind of like dinner plates, and it has a distinct polarity. It has a cis face, which is the receiving dock that gets packages from the ER, and it has a trans face, which is the shipping dock.
And in between those two faces, it does the addressing.
This is arguably its most important function, glycosylation. The Golgi adds specific complex sugar tags to the proteins as they pass through. These tags act like shipping labels or barcodes. A specific sugar pattern tells the cell's logistics system. This protein goes to the lysosome, or this protein needs to be embedded in the cell membrane, or this one gets exported from the cell entirely.
So without the Golgi, the mail would never get delivered to the right address.
It would be complete chaos. You'd have digestive enzymes floating freely in the cytoplasm, just eating the furniture.
Speaking of eating the furniture, let's talk about the lysosomes, the recycling center. Or maybe it's more like the stomach of the cell.
It's really both. Lysosomes are little vesicles that bud off from the Golgi. They're basically just little bags filled with powerful digestive enzymes called hydrolases enzymes that break down proteins, fats, and carbohydrates.
And there's a critical safety feature here that highlights our regulation theme perfectly.
The pH balance. The enzymes inside the lysosome are designed to only work at a very acidic pH, around 5.0. The rest of the cell, the cytosol, is neutral, around pH 7.2.
Why design it that way? Why not have enzymes that work at a neutral pH?
It's a brilliant failsafe. The lysosome uses proton pumps on its membrane to actively force acid inside itself, which activates the enzymes. But if the lysosome accidentally ruptures and spills those powerful enzymes out into the cytoplasm,
The neutral pH of the cell instantly deactivates them.
exactly. The acid spills, but the enzymes just turn off. The cell doesn't eat itself from the inside out. It prevents autolysis. It's a beautiful piece of engineering.
But sometimes the cell actually wants to eat parts of itself. There's a process called autophagy.
Self-eating. It sounds morbid, but it's an essential housekeeping process for cellular health. If a mitochondrion gets old and starts becoming inefficient, it can start leaking dangerous reactive oxygen species.
So it's a liability.
A huge liability. The cell recognizes this, wraps it in a membrane, and then fuses that package with a lysosome to have it digested. It recycles all the raw materials, the amino acids, the lipids, to build new things.
It's the ultimate form of sustainability. Nothing is wasted.
Right. And there is one more organelle that kind of looks like a lysosome, but it's very different. The peroxisome.
The hazmat team.
That's a great name for it. If lysosomes break down solid waste, peroxisomes deal with chemical waste. They contain a different class of enzymes called oxidases. They specialize in breaking down very long-chain fatty acids and detoxifying heavy chemicals like alcohol.
But the name peroxisome comes from a byproduct of its reactions, right?
Yes. The oxidative reactions that they run create hydrogen peroxide ERO as a byproduct.
Which is very toxic to the cell.
Highly toxic. So the peroxisome comes pre-equipped with a massive amount of a specific enzyme called catalase. The job of catalase is to instantly neutralize the hydrogen peroxide, breaking it down into harmless water and oxygen.
So it creates the poison and the antidote in the same room at the same time.
It has to. It's a perfect example of regulation containing the dangerous power of capacity.
Okay, moving on to section 4. We need a power source to run all these pumps, all these motors, all these factories. It's time to visit the power plant, the mitochondria.
The powerhouse of the cell. It's the biggest cliche in biology, but it is absolutely true. Guyton notes that for most cells, a full 95% of their energy comes from mitochondria.
Describe the structure for us, because in biology, structure almost always dictates function.
Right, so a mitochondrion has two membranes, a smooth outer skin and a highly folded inner membrane. These folds, which look like shelves, are called cristae.
Why all the folds? Why not just a simple bag inside a bag?
Surface area, it's always about surface area. The enzymes that actually generate the power, the machinery of the electron transport chain and oxidative phosphorylation, are embedded in that inner membrane. By folding it so intricately, you can pack way more enzymes into a very small space.
So more folds equals more power generation capacity.
Exactly. And there is something a little spooky about mitochondria that points to their evolutionary history, isn't there?
Yes, they have their own DNA. It's called mitochondrial DNA, or mDNA, and they reproduce independently of the cell.
Wait, so the nucleus doesn't give the order for them to divide?
Not directly, no. This
Yeah.
all points back to the endosymbiosis theory. We think that billions of years ago, mitochondria used to be independent bacteria that got swallowed by a bigger cell and just stayed.
Huh.
But functionally, this independence is crucial for our regulation versus capacity theme.
How so?
If a cell is under high energy demand, if it's using up ATP faster than it can be made, the mitochondria sense this local deficit in ATP, and they start replicating themselves to meet the demand.
Can we use a real-world example here? Absolutely. Think about the skeletal muscle cells in a marathon runner versus a couch potato.
Okay.
The runner's muscle cells are constantly screaming for energy. Over weeks and months of training, those muscle cells will physically increase their mitochondrial density. The cells themselves become darker and richer because they are packed to the brim with power plants.
And the couch potato cells.
Their cells say, hey, we don't need all this expensive machinery lying around, and the number of mitochondria actually drops. It's pure supply and demand. The capacity of the cell literally adjusts to meet the experienced need.
That is so cool. It's not just you get what you're born with. Your cells are actively renovating their internal architecture based on how you live your life.
Every single day. It's incredible.
Okay, section 5. Import and export logistics. Endocytosis and exocytosis.
Right, we've talked about the walls and the factories, but how do you actually move bulk material across the border? The key concept here is that the membrane surface is a continuous conveyor belt.
What do you mean by a conveyor belt?
Well, think about exocytosis exporting things out of the cell. You wrap a package, like a hormone or a neurotransmitter, in a little bubble of membrane called a vesicle.
Yeah.
That bubble then travels to the outer cell membrane and fuses with it to spit the package out. When it fuses, that bubble's membrane becomes part of the cell membrane. So you've just added surface area. The cell just got a tiny bit bigger.
So the cell should be constantly expanding.
It would be if you didn't have the opposite process, endocytosis importing things. When you bring something in, you pinch off a piece of the outer membrane to make an internal bubble. So you subtract surface area. The cell is constantly balancing this, adding and subtracting to keep its overall size remarkably stable.
Let's look at the mechanism of exocytosis, the export. How does a little bubble full of insulin that's inside a pancreatic cell know exactly where to go and when to pop?
It uses a sophisticated docking system. It involves proteins called snares. You have a V snare on the vesicle and a T snare on the target membrane, the cell surface.
So it's like a lock and a key.
It's more like a winch and a hook. They are designed to recognize each other and twist together. They coil up, pulling the vesicle forcefully against the membrane until the two lipid bilayers merge into one. This is often triggered by an influx of calcium ions.
And this is how our nerves release neurotransmitters to talk to each other.
The exact same mechanism. A nerve impulse causes calcium to rush in, the snare snaps shut, and the signal is released into the synapse.
Now flipping it, endocytosis, the import process. There are different ways for a cell to eat, right?
Right. There is phagocytosis, which literally means cell eating. This is for very large particles, like an entire bacteria or cellular debris. Macrophages and other immune cells do this. It's a very aggressive, violent act. The cell literally reaches out with extensions and engulfs its enemy.
And then there's panocytosis.
Cell drinking. This is the cell taking in very small sips of the extracellular fluid, mainly just to sample what's out there in the environment. It's not very specific.
But the most sophisticated one, the one that really just blew my mind with its elegance, is clathrin-mediated endocytosis. This sounds like something out of science fiction.
It is a beautiful piece of geometric engineering.
Okay.
So let's say the cell wants to grab a very specific molecule, say LDL cholesterol, the bad cholesterol. It has specific receptors for LDL on its surface.
Okay.
When LDL binds to that receptor, it triggers a protein on the inside of the cell called clathrin to start gathering at that spot.
Clathrin.
Clathrin is a protein shaped like a triskelion. It has 3 legs. These triskelions link together to form a geometric basket, like a geodesic dome, on the inner surface of the membrane.
So they physically force the membrane to curve inward.
Yes. As the cage assembles, it pulls the membrane into a deep pit. Then another protein called dynamin wraps around the neck of that pit, the stalk that's still connecting it to the outside. Dynamin squeezes like a lasso.
Snap.
Snap. It pinches the neck and cuts the balloon loose. And now you have a perfectly formed vesicle containing exactly what you wanted to import safely inside the cell.
That is just amazing. Now, we mentioned crawling earlier. Section 6, acting globally, locomotion and cilia.
How cells move from place to place.
We tend to think of sperm cells swimming with their tails, but most cells in our body don't have tails. And yet, your white blood cells can actively hunt down a bacteria through your tissues. How does a blob with no legs move?
Through amoeboid movement. And it is literally, as you said earlier, building the road in front of you while destroying it behind you.
Break that down for me.
Okay, step 1 is chemotaxis. The cell has receptors that can smell a chemical trail left by a bacteria. It knows which direction to go.
Okay.
Step 2. The cell rapidly assembles actin networks at the front edge, pushing the membrane forward in the direction of the smell. This extension is called a pseudopodium, a false foot.
So it grows a foot and points it towards its target.
Exactly. It then anchors that new foot to the surrounding tissue. Then, step 3. Myosin motors at the back of the cell contract and pull the rest of the cell body forward towards the anchor point.
It's literally dragging itself along by its bootstraps.
And then it dissolves the actin at the back and moves those building blocks to the front to extend the foot again. It is a continuous, relentless cycle of construction and demolition.
That is active hunting. It's incredible. Now, what about cilia? That's a different form of movement.
Very different. Cilia are these whip-like appendages that stick out from the cell surface, and they have a very specific internal structure called the 9+2 arrangement.
What does that mean?
It means they have a ring of 9 doublets of microtubules with 2 single microtubules running down the center. This is the axonome.
And they move?
The motile cilia do. They use dynein arms, the same motor protein from before, to physically bend the microtubules, creating a coordinated whip-like beating motion. You have billions of these in your airways.
And they're not for moving the cell, they're for moving stuff past the cell.
Exactly. In your lungs, they form a mucociliary escalator that is constantly beating upwards, pushing mucus and any trapped dust or bacteria up and out of your lungs so you can swallow it.
So if you smoke and you damage these cilia,
the escalator stops working, the mucus pools in your lungs, bacteria can grow, and you get chronic infections. That persistent smoker's cough is your body's crude attempt to mechanically clear out the gunk that the cilia can no longer handle.
there's a note here about primary cilia. These are non-modal. They don't move.
Yes. This is a relatively new and really exciting realization in physiology. It turns out that almost every single cell in your body has one single cilium that doesn't move. It just sits there.
And it acts as a cellular antenna.
It's an antenna. It senses fluid flow, chemical signals, and mechanical stress in the local environment. For example, in the kidney tubules, these primary cilia bend in the flow of urine.
And that tells the cell what's going on.
It tells the cell that everything is working normally. If those cilia are broken, which happens in certain genetic diseases, the cell goes blind to its environment. It panics, it doesn't know what to do, and it starts behaving erratically, often forming cysts. This is the basis of polycystic kidney disease.
It's incredible how much crucial function is packed into these tiny little structures. A broken antenna can destroy a whole organ.
It really is. It links molecular structure to large-scale disease absolutely perfectly.
Section 7. We can't talk about the cell without talking about the instruction manual. Synthesis and genetic control.
The central dogma. RNA makes protein.
We know the nucleus has the genes, somewhere around 20 to 25,000 of them. But the absolute key here, once again, is regulation. Not every gene is turned on in every cell. A kidney cell has the gene for insulin, but it keeps it locked up and silent.
Right. And it's all about access. DNA isn't just a loose string floating around in the nucleus. It's wound tightly around these protein spools called histones.
Okay.
If the winding is very tight, the gene is effectively off. The cellular machinery can't get in there to read it. If the winding is loose and open, the gene is accessible and considered on.
So a huge part of gene expression is just controlling how tightly those spools are wound.
To a very large extent, yes. And we have DNA regions called promoters and enhancers. These are like the volume knobs for gene expression. Proteins, called transcription factors, bind to these regions to tell the cell, hey, unwind this specific section. We need to make this protein right now.
And finally, this all culminates in the cell cycle, the ultimate decision for a cell to divide.
This is the biggest decision a cell can make: mitosis. And it is, as you can imagine, heavily regulated. Cells usually only replicate when they are signaled to do so by growth factors. And this is key. They stop when they get crowded. It's a phenomenon called contact inhibition.
They can literally feel their neighbors and they say, okay, the neighborhood is full, we should stop building.
Exactly. And when that regulation fails, when a cell ignores the stop signals from its neighbors and just keeps dividing and dividing despite being crowded, that is the fundamental definition of cancer.
It's a failure of social responsibility at the cellular level.
That's a perfect way to put it.
Wow. Okay, so putting this all together, we've gone from the fat sandwich wall to the taxol frozen highways, the acidic stomach, the self-replicating power plants, and the self-building roads.
It really is an ordered chaos, isn't it?
That's the outro theme here, a grand synthesis.
The cell is just a masterpiece of balance. It's constantly balancing capacity, all those mitochondria, the massive ER, with regulation, which comes from the genes and the receptors on its surface. It balances stability from the cytoskeleton with flexibility from the fluid membrane.
And the so what? Why does this deep dive into one tiny cell matter for someone listening who isn't a biologist?
Because every single disease you can name starts right here, at this level, a viral infection. That's a hijacker that has learned to use your endocytosis machinery and your ribosomes against you.
Cancer.
That's a mitotic regulation failure. Heart failure. That's often an issue of mitochondrial exhaustion. Type 2 diabetes. That's a receptor problem in the glycocalyx in the cell membrane.
So understand the micro logic of the cell is the only way a doctor can truly understand the macro failure of the patient.
Exactly. You simply can't fix the car if you don't know how the spark plugs work. This is where it all begins.
I want to leave our listeners with a final provocative thought. We've talked today about the cell as something that hunts, eats, defends itself, and communicates. It acts like an independent creature.
It really does, in many ways.
So if a single cell is a self-regulating entity, at what point does a collection of trillions of these cells become us? Is consciousness just the emergent hum of all these tiny cities trading information with each other? Are we just the aggregate noise of a trillion microscopic negotiations happening every second?
That is the ultimate question in biology, isn't it? And physiology can explain the mechanism of every single one of those negotiations, but it can't quite explain the magic of that final leap.
Something to mull over.
Yeah.
Thank you for joining us on this deep dive into the cellular city.
A pleasure, as always.
Check out the show notes for links to Ganong and Gaiden if you feel brave enough to tackle the original texts. Until next time, stay curious.